So guys, last time we discussed about the like Drosophila P element. Now what are controlling elements? So controlling elements was the term which was first used by Barbara McClintock and for transposable elements, and McClintock discovered two classes of controlling elements, which are AC and DC, AC and DS. Okay, AC is for activator and DS for dissociation. And she found that the DS elements could move in the presence of AC and they were unable to move alone. DS elements were unable to move alone, but DS can move in presence of AC. But AC is capable of moving alone. So that this product is transposes. So we can say that the product is the transposes. So that the activator protein which is when there, AC product when it is there, it could promote the movement of both AC and DS. So we can say that this product is transposes. And DS is what? DS is an insertion derivative of AC. DS is an insertion derivative of AC which contains the same spatial recombination sequences at its tips where the transposes produced by AC can act. Okay, so what is DS? DS is a deletion derivative. DS stands for dissociation, but it is a deletion derivative of AC which contains the same spatial recombination sequences at its tips. At its tips there is the same spatial recombination sequences where the transposes produced by AC can act. Okay, where the transposes which AC produces can act at the tip. So DS lacks the transposes gene, it cannot promote its own movement and it is dependent upon AC. So DS is non-autonomous controlling element, whereas AC is the autonomous controlling element because AC can encode its own transposes. And in addition to AC DS element, there are several other autonomous and non-autonomous elements are present, which is reported in maize and some other organisms. So next we talk about retrotransposons. So as discussed, maximum number of transposons which are found in eukaryotes are retrotransposons. Okay, and retrotransposons have an RNA intermediate which utilizes a reverse transcriptase, and these mobile elements, they are divided into two major categories: viral and non-viral retrotransposons. What are viral retrotransposons? Viral retrotransposons, they are similar to retroviral DNA. Okay, how? Because in retroviral DNA, viral retrotransposons, like whatever happens in retroviral DNA is that they are flanked by 250 to 600 base pair long terminal repeats. Okay, so LTR is there at the flanking region, which is 250 to 600 base pair long, and like retroviruses, they encode reverse transcriptase and integrase. So viral retrotransposon, they are kind of retroviruses. In retrotransposons, there is a flanking region at both ends known as LTR, which is long terminal repeat, and in addition it has the reverse transcriptase and integrase element. So retroviruses, they are what? They are single-stranded RNA genome, and what happens that in the host cytoplasm, retroviral genomic RNA, first it is reverse transcribed, as you can see in the picture, that first it is reverse transcribed into double-stranded DNA, then it integrates into the host genome, and the double-stranded DNA is inserted into the host genome by the transposition mechanism, which like generates short direct repeat of target DNA. Okay, so you can see in the picture that first of all there is reverse transcription and integration. First, in the first case it is GAG, POL and ENV. In the second case you can see that the GAG, POL and ENV, these are flanked by LTRs. When it is integrated, you can see that there is direct repeat, then LTR, and in the middle there is the gene. Okay, So double-stranded DNA is inserted into the host genome by transposition-like event, which generates short direct repeat. So after transposition, there is short direct repeat. Okay, and a retroviral sequence that is integrated, it remains as endogenous provirus. Okay, provirus, like it is integrated, like the repeats are not integrated, but the gene is integrated, which contains GAG, POL and ENB. So this viral retrotransposon, they move in the genome by being transcribed into RNA. Okay, so first of all, there is reverse transcription. When the process started, there is reverse transcription. Then they are integrated, because DNA will integrate DNA only. So, but when they move into the genome by being transcribed into RNA, which then undergoes reverse transcription and then integrates into the host cell chromosome. And this viral retrotransposons, they are abundant in yeast, example TY elements, and in Drosophila it is copia elements. So retrotransposon in Drosophila is copia, and in yeast it is TY. So this is a general structure of viral retrotransposon, where you can see direct repeat, then LTR, then there is protein coding sequence. Okay, so like other mobile elements, there is short target site repeats at each end, and the protein coding region, here it is constituting the 80% or more of a retrotransposon, and this protein coding region, this encodes reverse transcriptase, integrates in some virus specific proteins. So what is the first step in retrotransposition? The first step is the synthesis of RNA copy. Okay, synthesis of an RNA copy of the inserted viral retrotransposon by RNA pol 2. So first step is reverse transcription, where RNA to DNA is formed, DNA is integrated. Once DNA is integrated, then again RNA copy is formed by RNA pol 2. And this transcript encodes a reverse transcriptase enzyme that makes double-stranded DNA copy of the RNA molecule via DNA-RNA hybrid intermediate. So what happens is that in the first step, it is a synthesis of an RNA copy of the inserted viral retrotransposon. First RNA copy will be formed, okay, and then there is a reverse transcription that makes a double-stranded DNA copy. First there is RNA copy, then double-stranded DNA copy, okay, via a DNA, like whatever happens in like replication, DNA RNA hybrid is formed, and this analogy will continue as a linear DNA molecule uses an integrase. So their next step will be integration. First step is RNA copy formation, then it is DNA formation, then it is integration by integrase enzyme. Okay, and integration is into the selected, randomly selected site on the chromosome. It's not specific. Now this was viral retrotransposon. What about non-viral retrotransposon? Non-viral retrotransposon do not contain long terminal repeat, and they have 80 rich stretch at one end. At one end there is 80 rich strand, and in mammals, non-viral retrotransposon are the most common type of mobile element. Okay, in animals it is the most common non-viral kind of retrotransposon, and the most abundant non-viral retrotransposon, they are line and sign. Okay, they are retrotransposon, but they are non-viral, and they are line and sign. So what is line? They are long interspersed nuclear element, and what is sign? They are short interspersed nuclear element. Now what are signs? Signs are around 100 to 400 base pair sequences which does not contain any gene. Okay, they are just a sequence, does not contain any gene. They are transcribed by RNA polymerase 3, and the most common signs in humans, they contain a site for restriction enzyme ALU1, and they are called ALU sequences. So sign is not containing any gene. What sign contains? They contains restriction enzyme for ALU1. So they are known as ALU sequences. And these signs, they are scattered throughout the human genome, which constitute around 10% of the total DNA. Okay, 10% is too less. They are scattered. And a related transposable element B1, it is found in the mouse genome. So you can say kind of sign in mouse is B1. Okay, and both of these transposable DNA sequences, they are thought to have evolved from the gene for 7SL scRNA, a non-coding RNA involved in polypeptide movement of the involved in movement of polypeptide in the organelles. So you can understand that this is a typical structure of a line element, and the shorter open reading frame ORF1, which is about 1 kb in length, it encodes an RNA binding protein, and the longer ORF2, which is about 4 kb in length, it encodes a bifunctional protein with reverse transcription and DNA endonuclease activity. So one is encoding RNA binding protein, another is encoding what? Bifunctional protein. Okay, bifunctional have reverse transcription and endonuclease activity. So the most common line element, it constitutes the L1 line family. Most common line element constitutes the L1 line family. Okay, and most common sign in mouse is B1. Okay, and sign have ALU element. So L1 line family, there are around 6 lakh copies of L1 elements occur in the human genome, which is about 15% of the total human DNA. So line are more than sign, and line are flanked by short direct repeats, which are the hallmark of mobile elements. So whenever you will understand about mobile elements, it is the direct repeats, and the sequence contains two long open reading frame. Okay, so line structure, if you try to understand, there are two direct repeats, which is a characteristic of the transposable element or mobile element, and this line also have a characteristic of containing two open reading frames, one encoding RNA binding protein and another encoding the bidirect kind of bifunctional protein, which is reverse transcriptase. So there is a transpositional site-specific recombination by non-viral retrotransposon. You can see in the picture that L1 element in chromosome, L1 RNA synthesis first occurs. Okay, there is the L1 RNA synthesis, then the synthesis of reverse transcriptase endonuclease and binding with L1 RNA. So first of all, L1 element in chromosome, okay, which is AT rich, you can see L1 RNA synthesis will occur, single stranded L1 RNA will be formed, then the binding of reverse transcriptase endonuclease and binding with L1 RNA. So L1 RNA, here it will reverse transcriptase will be synthesized and it will bind to L1 RNA. Okay, then there is a cleavage of one strand of target DNA, one strand will be cleaved, and then DNA primed reverse transcription will occur, there will be second strand formation and new DNA copy is formed at the new position. So basically what? New DNA copy is formed in case of line elements, which is what? Which is, it is helping in transposition. So from L1 element, there is a synthesis of RNA, L1 RNA synthesis, and then there is the reverse transcriptase endonuclease, which will bind to L1 RNA, and one strand will be cleaved. Okay, one strand will be cleaved, and then it will there is DNA bind reverse transcription will occur, and there is a formation of second strand. So what difference is there between viral retrotransposon and non-viral retrotransposon? The enzymes which are required are moreover same in both. In non-viral, endonuclease is required, which needs to nick the strand. LTR is not present in non-viral. Target repeat is a bit longer in non-viral, and common types of viral retrotransposon, which is TY element in yeast, copia in Drosophila, and non-viral retrotransposon, which is L1 element, and B1 is sign, which is found in mouse. So This was the general idea about the sign element and bacteriophage P element and ACDS element. Now coming to what is gene. So a gene is what? Gene is where information is stored, okay, and it's double helix functional structure, and they are transcribed into mRNA, which is translated into proteins, and the genes include all the DNA sequences, okay. So all the messages is there in the gene, and it has many regulatory sequences also responsible for uh, the transcription at proper time and appropriate cell type. So number of sizes of the gene. So for several organisms the sizes are different. In the bacterial genome it is the number of gene is proportional to the genome size in case of bacteria, and the bacterium with the smallest known genome is M genitalium, which is just 470 genes, and in eukaryotes it varies from 6000 to 40,000 genes, okay. But in eukaryote you cannot correlate the number of the genes with genome size, okay. Some genes are present in more than one copy or related to one another, and there are different types of genes which is less than the total number of genes. So M genitalium having least is 470, H influenzae 1743 genes, subtilis 4100, E. coli 4288, S. cerevisiae yeast which is 6034, Drosophila 12,000, C. elegans 19,000. So it varies greatly, and there are genes which contains more than 2 million nucleotide pairs. There are genes which contains around 1 lakh nucleotide pairs in length are common. So there are short segments also in the gene which are for regulatory roles known as introns. The coding part is known as exons, and the RNA molecule which is known as a primary transcript, which is synthesized from such a gene, is processed to remove the intron sequences, okay. There is a splicing mechanism which is used to remove the intron sequences, and introns were first discovered in 1977 by Philip Sharp and Richard Roberts. And introns, they are present in most of the genes in eukaryotes, and they are not universal, and many genes like interferons, histone genes, ribonuclease genes, heat shock protein genes, G protein coupled receptors, they all lack introns. There are many genes which lack introns, so introns are not actually required for the function. So they are not found in most of the genes in simple eukaryotes like yeast, but uh, rarely it is present in prokaryotes. And exons and introns are present alternatively, and the overall length of the gene is determined largely by the introns. Introns are quite longer and larger than the exons, and they can range from 50 nucleotides to 8 lakh nucleotides, and they are usually exons are usually short for around 150 nucleotides in average. So number of exons, size of the gene, and average size of intron is mentioned for some proteins. Like for insulin it is just 1400 base pair, where exons is 3 and intron is 480. Beta globin gene is 1.6, number of exon is 3, intron is 490. Serum albumin 18 kV, exon 14, intron is 1100. CFTR size is 250 base pair, exon is 27, average size is 9100. Titin is 283 kb, number of exon 363. You can see there are so number of, it's not related to the size, okay. Average size of intron is 466. Dystrophin is 2400, exons is 79, size of intron is 30,000. So introns have been reported in both prokaryotes and eukaryotes, but rarely in prokaryotes, and They may contain introns for nuclear genes for proteins, nucleolar genes coding for rRNA, and genes coding for tRNA. And introns are also found in mitochondrial genes and chloroplast genes as well. As we have studied, there is organelle DNA also in addition to nuclear DNA. And the most common type of introns are mentioned here in this table, and they do not code for proteins, but certain type of introns like group 1 and group 2 classes, they contain open reading frames which are translated into proteins, and expression of the proteins allows the introns to be mobile. So what are the types of most common introns? It is GUAG intron, which is found in eukaryotic nuclear pre-mRNA. AUAC also found in eukaryotic nuclear pre-mRNA. Group 1 is also found in eukaryotic nuclear pre-rRNA, organelle RNA and some prokaryotic RNA. Group 2 is found in organelle RNA and some prokaryotic RNA. Pre-tRNA introns is found in eukaryotic nuclear pre-tRNA. Archaeal introns are various kind of RNA. Okay. So there are several evidences where introns exist or does not exist in eukaryotic genes. So early evidence about the existence of introns in eukaryotic genes, it was provided by R-loop technique. So if it is asked that which technique is used to find out whether introns exist or not, it was R-loop technique, in which a base paired complex between mRNA and DNA molecule is visualized in the electron microscope. So between mRNA and DNA molecules, the base pairs are visualized in electron microscope, and when purified, say for they have studied ovalbumin mRNA. So when purified ovalbumin mRNA preparation, it is annealed to double stranded DNA. So mRNA is what? mRNA, you can say that when only exon is there, no intron. So when this is annealed to double stranded DNA molecule in a suitable condition, when the gene that encodes the mRNA, the RNA can displace a DNA strand wherever the two sequences match and form a region of DNA RNA helix. So wherever there is the match, okay, that region will match and the unmatched region will form a loop, okay, which is the intron loop. Okay, so RNA can displace a DNA strand. Okay, RNA is displacing the DNA strand. RNA is displacing the DNA strand where there is a match, and the unmatched region will be visible as a large loops of double-stranded DNA. Okay, so when double-stranded DNA was taken, mRNA replaces one of its strand, and somewhere where there is no match, there is a loop of double-stranded DNA was formed, which represent the introns. So this R-loop technique, in which a base paired complex between mRNA and DNA molecule, it is visualized in the electron microscope, and when this single-stranded mRNA preparation, it is annealed in a suitable solvent to a clone double-stranded DNA molecule containing the gene that encodes the same mRNA. The RNA can displace a DNA strand wherever the two sequences match and form region of RNA-DNA helix. So region of DNA where no match to the mRNA sequence is possible, they are visible as large loops of double-strand DNA, and each of these loops represent an intron in the gene sequence. So this was about kind of exons and introns and gene and the transposable elements, retrotransposons. In the next video, we will start with gene duplication.